What is neurotransmission? Neurotransmission is crucial for brain function, allowing neurons to communicate via chemical signals. But what happens when this intricate system is disrupted by brain cancer? First, let's understand the basics of neurotransmission. Neurons, the brain's communication cells, talk to each other using chemicals called neurotransmitters. Key players include dopamine, serotonin, and glutamate. Each neurotransmitter has a unique chemical structure. Dopamine, for instance, contains a catechol group and an amine group, making it a catecholamine. Serotonin has an indole ring structure, and glutamate is an amino acid with a carboxylate side chain. How are these neurotransmitters synthesized? Dopamine is made from the amino acid tyrosine through a series of steps involving enzymes like tyrosine hydroxylase and DOPA decarboxylase. Serotonin is synthesized from tryptophan via tryptophan hydroxylase and aromatic amino acid decarboxylase. Glutamate is produced from glutamine by the enzyme glutaminase. Neurotransmitters are stored in vesicles at the synaptic terminal. When an electrical signal reaches the end of a neuron, calcium ions enter the cell, triggering vesicles to fuse with the membrane and release neurotransmitters into the synaptic cleft. These neurotransmitters then bind to specific receptors on the neighboring neuron, transmitting the signal. In brain cancer, particularly gliomas, this delicate balance is disrupted. Tumors can alter the level of neurotransmitters and the expression of the receptors. For example, gliomas often release large amounts of glutamate, causing excitotoxicity, where excessive activation of glutamate receptors leads to neuronal injury and death. These disruptions can result in various neurological symptoms such as cognitive deficits, mood changes, and seizures. The chemical imbalance caused by the tumor affects neurotransmitter synthesis, release, reuptake, and receptor signaling pathways. Understanding the chemistry of neurotransmission helps us develop new treatments. Researchers are creating targeted therapies that restore neurotransmitter balance or block harmful signaling pathways. One approach involves using NMDA receptor antagonists to prevent excitotoxicity caused by excess glutamate. By focusing on the chemistry of neurotransmission, we can develop innovative treatments that may improve the lives of those affected by brain cancer.